Good morning, dear students. Today we are going to begin with your biology practicals. The very first practical that we are going to discuss in the zoology section is the practical number 4. The aim of the practical is to study the discrete stages of gametogenesis in mammalian testis and ovaries. So in this practical, we are going to study about the transverse section of testis and of the ovaries. Now we already know that whenever we take a transverse section of the testis or ovaries, we will be able to observe the cells in different stages, in both within the testis as well as in the ovaries. Now this transverse section is mainly seen for the study of the process of gametogenesis, that is formation of the gametes. Within the testis there is formation of the sperms, and within the ovaries there will be formation of the egg, and these processes are known as spermatogenesis and oogenesis respectively. In this practical we have to study only the permanent slides or the charts that are being given to you. So for the practical purpose, a permanent slide will be given and you will have to just identify the different stages of the cells that are observed in testis and in the ovaries. So permanent slides will be given and you do not have to prepare any slides for this practical. So first of all, we begin with the principle of this practical. We already know that in male and female organisms, the gamete formation takes place in the gonads. So gonads are those primary sex organs in which the gamete formation is going to occur. These primary sex organs will be testis in the males and ovaries in the females. The process of formation of gamete, जो gamete बनने की procedure है, this procedure is known as gametogenesis. Or whenever the gametogenesis occurs, it involves the meiotic cell division, that is the reductional cell division in which the chromosomes number are going to reduce to half. And therefore, formation of sperms is called spermatogenesis and it takes place in testis. Formation of the egg is known as oogenesis and it takes place in the ovaries. The requirement of this practical, for this practical you will be requiring the permanent slide or maybe a chart can be provided to you, right? Some of the times instead of permanent slides, a chart is being provided and you have to identify it and you have to label it or you have to answer any question related to this practical. You do not have to perform this practical, right? You have to simply observe the pre-prepared permanent slides in the microscope. So permanent slides will be given, that is transverse section of testis and ovaries will be given. You require a compound microscope, a cleaning fluid just for cleaning the lens. So before you observe it in the slide, it is very important for you to clean the lens. You will have to observe this both in low power also and then high power objective lens. Now what will you observe? So in the procedure simply you have to first of all take a slide. You have to place it in the microscope. You have to attach the clips to it. You have to clean it with the cleansing paper, right? That is cleansing fluid that will be used for cleaning the lens. And you have to simply observe what do you see in the transverse section. So in the transverse section of the testis, what are we going to observe? In the transverse section, first of all, the outermost layer of the cells that will be observed is called germinal epithelium. मतलब हम जब टेस्टिस देखते हैं तो टेस्टिस के अंदर होते हैं सेमिनिफेरस ट्यूब्यूल्स. यही सेमिनिफेरस ट्यूब्यूल्स में हमें देखने को मिलेंगे the first types of cells which are called the germinal epithelium. So outermost layer will be of germ cells. Just below the germ cells, you will be able to observe different cells beginning from periphery to the lumen. Periphery से लेके lumen तक आते आते अलग-अलग developmental stages में cells देखने को मिलेंगे. So the first type of cells that will be observed are spermatogonia that are formed by numerous mitotic divisions, right? Germ cells will be dividing by mitosis and they will be giving rise to spermatogonia. Out of them, some of the spermatogonia are going to develop into primary spermatocyte. Means a spermatogonia which will enlarge in its size is going to form a primary spermatocyte. This primary spermatocyte undergoes meiosis 1 to give rise to the secondary spermatocyte, and after the secondary spermatocyte which undergoes second meiotic division to form the spermatids, and these spermatids undergo several changes for a very long time and change into spermatozoa. तो पेरिफेरी से लेके ल्यूमेन तक आते आते आपको अलग अलग डेवलपमेंटल स्टेजेस में सेल्स देखने को मिलेंगे. जरूरी नहीं है कि हम क्लियरली सारे ही सेल्स को आइडेंटिफाई कर सकें, but our attempt will be that we are able to observe all the cells in the region of the seminiferous tubules. Between will be the Sertoli cells, that will be slightly elongating cells which are also known as nursing cells, and between the two seminiferous tubules there will be presence of the connective tissue and blood vessels which will also have the presence of another type of cells called Leydig cells, right? And Leydig cells will be the one which will be producing the male hormones testosterone. So in the transverse section of testis you will be able to see different cells in different developmental stages. Similarly, if we take the transverse section of ovary and observe it in our microscope, what are we going to observe? So in the ovary you are going to observe different developmental stages of the cell. Initially there will be presence of the primary follicles. These primary follicles will be changing into secondary follicles. The secondary follicles will be having the presence of, that is, primary oocyte within them. Okay, so there will be presence of a primary oocyte in it. Then after you see the presence of tertiary follicles. Now the primary oocyte has divided and it has formed the secondary oocyte only. So there will be formation of the tertiary follicle with the secondary oocyte, and it will be characterized by presence of a fluid filled cavity which is called antrum. The largest size follicle that you will be able to observe is the Graafian follicle. मतलब आपको शायद सारे ही developmental stages अच्छे से ना देखने को मिले, but you will be able to observe the stage which one? Graafian follicle, because size of Graafian follicle is slightly larger and it is visible in the microscope. So you will be able to observe this structure which is called Graafian follicle. The cavity within it is called the antrum, and the cell that is present within it is the secondary oocyte. The Graafian follicle will break under the presence of the LH and there will be resulting into ovulation. Ovulation मतलब egg जो है that is going to be released out. After this you might be able to observe the presence of corpus luteum, because once the egg, that is the secondary oocyte, gets released out, rest of the Graafian follicle changes into the corpus luteum, and the broken down corpus luteum will be observed in the form of a small scar which is known as corpus albicans. So that is how different stages will be observed in the ovaries. I am again saying that we are here observing permanent slides. So it might not be compulsory for you to observe each and every cell in it, but you can at least detect some of the stages, like you can observe Graafian follicles over here, you can observe germ cells and spermatogonia over here. So that will be observed in the microscope. So let us now see that how you should observe within a microscope. For observing the permanent slide for the mammalian testis and ovaries transverse section, we require a compound microscope. We require a cleaning paper for cleaning the lens. So we have to take care that before observing anything, first of all, with the help of a cleaning paper, we will have to clean the lens that is present at the top, as well as we will have to clean the objective lenses, because we are going to observe in both high power as well as in the low power. Then after, what we have to do is, we have already a pre-prepared slide. So we have to place this slide in the microscope, place a cover slip on it which will be already present, as we have a permanent slide for the observing the section of testis and for the ovaries. So we will have to place it over here, right? We have to attach the clips, and then after, with the help of the fine and the coarse adjustment screw, we will have to adjust it in such a way that we can observe the completely all the cells that are present in the seminiferous tubules as well as in the ovaries. So there will be two particular slides that will be given to you, and then after, you have to observe them with the help of the microscope. First of all, you will be observing it in the low power, and then after you will be shifting it to 45x. In order to observe it at 100x, we require oil immersion lens. That is, there should be presence of, that is, you have to take an oil immersion by which we can place a drop of it, and then after we will be observing in this microscope. In this microscope, you will be observing the section of the ovary, that is the egg cell like this. So we will be observing the different developmental stages, where we have seen that there will be formation of a mature follicle, that is a Graafian follicle will be forming, which will be having the presence of the antrum, and then after we can see sometimes the ovulated secondary oocyte that has been released. Corpus luteum and degenerating corpus luteum can be observed in this slide. So different stages can be observed. You have to identify them and describe them.